Today we're going to be talking about topic 11, which has to do with the mitochondria and cell respiration. With cell respiration, how do we get our energy? We get our energy from food. So cells use all different types of molecules of food, so fats, proteins, and carbohydrates. Each one of them gives us energy a little bit differently. So each one of these energies is stored in each molecule, and depending on their structure, depends on how their energy storing bonds are and how they differ. So cells break down food molecules over time and use the energy stored in the chemical bonds to produce compounds such as ATP that power the activities of the cell. So carbohydrates tend to give us food energy really quickly. Proteins kind of spread it out a little bit, and then fats usually give energy very quickly as well, but then they later on get stored into our body. What is cellular respiration? It's the process that releases energy from food in the presence of oxygen. So you take in food, you eat it, you breathe, cellular respiration is happening. So if oxygen is available, cellular respiration will happen. This is basically the chemical formula for cellular respiration. It's oxygen and glucose, and it makes carbon dioxide and water and energy. Now, if you read this backwards, then it's photosynthesis, because photosynthesis and cellular respiration are mirror images of one another. So there's three main stages of cellular respiration. There's glycolysis, there is the Krebs cycle, also known as the citric acid cycle, and then there's the electron transport chain, which we can also call the ETC. Pathways of the cellular respiration that require oxygen are called aerobic, which we've talked about aerobic before. Aerobic means that it's in the presence of oxygen and it uses energy. So Krebs cycle and the electron transport chain both take place in the mitochondria, and they both require oxygen. So they're both aerobic. Glycolysis is anaerobic. Remember, anaerobic means it does not require oxygen. So glycolysis happens in the cytoplasm of the cell, which is like the jello portion of the cell. So it's out of the mitochondria and it's anaerobic. The relationship between photosynthesis and cellular respiration, like I said before, they're mirror images of one another. So while photosynthesis takes out carbon dioxide and water, cellular respiration is going to produce it. While photosynthesis makes sugar and oxygen, cellular respiration is going to take that in. So they're mirror images. What happens during the process of glycolysis? I'm going to show you a recording of me drawing and explaining this. Hopefully that will give you a good understanding of it. So we're going to be talking about glycolysis right now. Glycolysis means that glucose is splitting. So glucose is normally a 6-carbon sugar. Six-carbon sugar. And we're going to split it. In order to split it, we need energy. Energy is ATP, which I spoke about earlier. So we're going to use 2 ATP to split this. So 2 ATP. So originally this had phosphates attached to both sides, and then it's going to use 2 ATP next to split into 2 3-carbons. In order to split, it has to give up those phosphates for a second. It's only going to give it up for a short amount of time, and then it's going to get them back later. So it's going to go from 2 adenosine triphosphates to 2 adenosine diphosphates. Tri means 3, di means 2. So this is going to become, which looks like the word poop, but it's not. Those are the 2 phosphates attached to both ends. And then we're going to have 3 carbon and 3 carbons. So it's split. So glucose got split. It basically got karate chopped in half. So 3 and 3. And then it has the 2 phosphates at the end. Now we're going to be going to the next part of this. So once it's split, it's still not what we need. So what we're going to do is we're going to have NAD+, which is an electron carrier, come around, and it's going to pick up hydrogens and electrons from here. And it's going to take them to the electron transport chain, which we're going to call the ETC. Then, while that's happening, ATP is going to come around and it's going to take its phosphates back and 2 more. So it's going to get rid of these. And it's going to be we're gonna have 4 ADP. It's gonna take away those phosphates, and it's gonna become 4 ATP. And we're gonna have pyruvic acid here. This is gonna be pyruvic acid. So the whole point of this glycolysis is to basically make pyruvic acid. So we're just kind of shifting things around and getting rid of certain things until we get pyruvic acid. At the end of this, we have pyruvic acid and we have 2 ATP made. So 2 ATP are made. Oops. 2 ATP are made.
So I know my drawing was a little bit crazy. Here is a image directly of it. You can see the process happening here as well. So like I said before, glucose means split. You're trying to split it. You're going to give 2 ATP. Not you, you're going to give. 2 ATP are going to be deposited, and afterwards there's going to be 4 ATP made. So this is basically the same idea of you lending somebody 50 cents and you want interest back, so you get back $1. You didn't actually earn $1, you only earned about 50 cents of it. So same idea is happening here. 2 ATP are deposited, 4 ATP are given back, then you only made 2 ATP. NAD plus is an electron carrier, and it's going to take a pair of hydrogen and high energy electrons, and it's going to become NADH, and that's going to go to the ETC. Glycolysis happens really fast because it's anaerobic, so it's going to make energy. When the energy demands of the cell suddenly increase, it's going to happen very quickly. During the Krebs cycle now, I'm going to have another crazy drawing, and this one's going to be even crazier. You're going to see the process of the Krebs cycle happening, and I'm going to explain different parts of it. Hopefully you'll understand it. If not, harass Samayoa and I and talk to us and ask us a bunch of questions.
So now we're going to be using that pyruvic acid for the next part of the cycle. So this is going to be the Krebs cycle or the citric acid cycle. They're both the same thing. They're just 2 different names to it. So that pyruvic acid is going to come around. Remember that we made 2 pyruvic acids. So here in this next part, one of those pyruvic acids is going to come in. It's a 3-carbon. It's going to be right there. And it's going to go into the mitochondria. This is the mitochondria. And that 3-carbon, as soon as it comes in, it's going to lose one of the carbons. So one of these carbons is going to go away, and I like to call it CO2 bi, because it's a byproduct, so it leaves. So CO2 is going to go away, and whenever a carbon dioxide leaves, NAD plus is going to come around, and it's going to take a hydrogen and electrons, a pair of electrons, and it's going to go to the electron transport chain. So now, since that carbon dioxide left, we are left with 2 carbons here. In the next part, we're going to have an enzyme called acetyl-CoA that all it's going to do is it's just going to prep that 2-carbon to attach to the 4-carbon that was here before. So it's going to attach them together, and it's going to make a 6-carbon right there. So that 6-carbon is going to be citric acid. So that's citric acid. So that 4 from the end of the cycle is going to attach to the 2 here. As a result, to the acetyl-CoA, it's going to attach and make 6. So in the citric acid cycle, we're going to have one of the carbons once again disappear. CO2 bi. And now we have 5 carbons, and every single time that carbon dioxide leaves, NAD plus comes around. It's going to pick up hydrogens. So we have that happening right there. And then we're going to have, once again, right after that, another carbon dioxide leave. And once again, we're going to have NAD plus come around, make NADH. And then we're going to have that 4 carbon, it's going to take, ADP is going to come around, it's going to take a phosphate that was there. And we will have 1 ATP. And we're going to have another 4 carbon right here. And we're going to have FAD, which is another electron carrier. It's going to be FADH2. Come around until we get back to the beginning where we have one more NAD plus come around. And it becomes NADH. And it's going to take the electron again. So at the end of this, what did we end up with? And remember, this whole cycle happens twice. So we had 1, 2, 3 carbon dioxide to leave. We had 1, 2, 3, 4 NADHs, and we're going to have 1 FADH2. But in reality, since we did this twice, we're actually going to have 6 carbons leave, 8 NADHs, and 2 FADH2. And we also have 2 ATPs made. I forgot to add the ATP. So 1 got made there. So we have 2 ATPs.
Hopefully you understood my recording and you get to see it all here in an actual drawing in the mitochondria. But hopefully you still understood exactly what I wrote. If not, once again, like I said, ask Samayo and I questions. But here's a better image of it. You get to see the carbon dioxide leaving. You see the NADH going to the electron transport chain, FADH2 going to the electron transport chain, ATP being made here. And you get to see as well that how many are made. So you get 6 carbon dioxides that leave, 2 ATP are made, 8 NADHs, and 2 FADH2s. All this happened because there was 2 pyruvic acids, so this process happened twice. The electron transport chain is going to use these high energy electrons now. So all those hydrogens and the electrons that came from the electron transport chain, from the electron. Now we're going to be seeing all those high energy electrons from glycolysis in the Krebs cycle come to the electron transport chain. All those hydrogens and high energy electrons are going to be dropped off here. And what they're going to do is all those hydrogens are going to go into this intermembrane space, and there's going to be a buildup here of hydrogens. Now, cells don't really like it when there's a buildup of something. They like to have a balance because of homeostasis. So all these hydrogens are going to kind of want to go here. Now, there's only one pathway that it can go through here, which is through the ATP synthase. This is a one-way. All of these are one-way. So there's 3 different channels for them to go to this area. There's only one channel for them to go through here. Now, the good thing about ATP synthase is it's going to make ATP. And I'm going to show you a video right now of what that process looks like.
Of hydrogen ions on the 2 sides of the membrane. This difference is a hydrogen ion, or proton, concentration gradient. The energy associated with this gradient is used to synthesize ATP from ADP and PI. This occurs at the ATP synthase complex. One hydrogen ion enters the ATP synthase complex from the intermembrane space, and a second hydrogen ion leaves it on the matrix space. The upper part of the ATP synthase complex rotates when a new hydrogen ion enters. Once 3 protons have entered the matrix space, there is enough energy in the ATP synthase complex to synthesize 1 ATP. In this way, the energy in the hydrogen ion gradient is used to make ATP. Now let's watch the process again. Notice how the proton enters the ATP synthase and exits into the matrix space. Once 3 more hydrogen ions have crossed the membrane, another molecule of ATP will be made. In this example, the hydrogen ion gradient is large enough to produce 6 ATP molecules. Please watch as the remaining ATP molecules are synthesized.
So as you saw with the video, all that hydrogen had built up here in the intermembrane space, and in order for it to pass through, it had to go through the ATP synthase molecule. So 3 hydrogens are enough energy to build up to make ATP, from ADP to ATP. And the ATP synthase is right here. So that's the only one-way path through from the hydrogens here to here. So each rotation is going to build up enough energy. How much energy does cellular respiration generate? So together, glycolysis, the Krebs cycle, and the electron transport chain release about 36 molecules, all 3 of them together. Now, depending on what book you read, depends on how much it actually says. So glycolysis always produces 2, Krebs cycle / citric acid always produces 2, and then the electron transport chain will usually produce between 30 to 32. So that's this number here for like the entirety can range between 34 to 38 is the numbers I've seen. So the cell can generate ATP from about any source. We've just modeled it around glucose. So when you don't use cellular respiration, we use something called fermentation. Now, fermentation can make 2 different kinds of things. One is lactic acid fermentation, which happens with us, and then there is alcoholic fermentation. Alcoholic fermentation is what's used to make beer and wine. Lactic acid fermentation is what we undergo when we need short bursts of energy. So if we need to do a quick weightlifting or if we need to do a quick sprint, we will undergo lactic acid fermentation because it will use whatever ATP is already in our muscles, and it'll use lactic acid fermentation as another way to produce that ATP. So with alcoholic fermentation, like I said, they'll use yeast and other microorganisms to undergo that fermentation process to produce alcohol. So it's used in alcoholic beverages, and it's also used in bread dough to help it rise. You don't have to memorize this equation. You just need to know the differences in both fermentations and that they are not in the presence of oxygen. Notice there's no oxygen present here. And they are usually done as quick energy. So how does the body produce ATP during different stages? For the quick burst of energy, it'll use the ATP already in the muscles, and then it'll use lactic acid, like I said. Any exercise for longer than 90 seconds, so when you're doing like a full hour workout, or if you're doing like sprinting or running for long distances, you're going to undergo cell respiration. Lactic acid fermentation can supply enough energy to last about only 90 seconds. Now, all the extra oxygen that's required to get rid of the lactic acid is going to be the issue. So sometimes we don't get rid of all the lactic acid buildup, and that causes the soreness in our muscles.